All right, get ready, everyone, because we are diving deep today into the world of inhibitory neurotransmitters.
Specifically,
Oh, yes.
GABA A and glycine receptors.
Oh, yes.
Those essential breaks that keep our brains from going haywire.
Yeah, day 22.
Of this exploration.
It's exciting.
And we are getting down into the nitty-gritty.
Absolutely. We're focusing on how these receptors
They work,
how they function.
So both GABA A and glycine receptors are these really interesting structures.
They are
ligand-gated.
Ligand-gated.
Ion channels.
Yeah, so they're kind of like these specialized doorways.
Yeah, think of it like doorways.
On the neuron surface.
On the surface, controlling the flow of ions. Like chloride.
Like chloride.
Exactly.
So when GABA or glycine
are inhibitory ligands.
Bind to their receptors.
It's like they're holding the door open.
Okay, so they're holding the door open for these chloride ions.
Yes, for the chloride ions to rush in.
Just to come on in, but why is chloride so important?
That's a great question.
How does
that influx actually inhibit?
Yeah, inhibit the neuron's activity.
So every neuron has this certain electrical charge, right? And to fire it needs to reach a specific threshold.
Okay, to send a signal.
Exactly. Chloride ions.
Yes.
They carry a negative charge. So when they flood into the neuron.
Okay, through these open channels.
Yes, it makes the inside more negative.
More negative, okay.
It shifts the electrical balance, you see.
I see.
Making it harder for that neuron to reach that firing threshold.
So it's kind of like moving the finish line
yes.
further away in a race.
Further away, exactly.
So the neuron has to work harder. To generate a signal.
That's a great analogy.
So this process is called?
Hyperpolarization.
Hyperpolarization.
That's the key to how GABA and glycine exert
their inhibitory effects.
Inhibitory effects, they're dampening.
Okay.
The neuron's excitability preventing it from firing.
Too easily?
Too easily.
So we know that these signals are really crucial.
They are.
For keeping a healthy balance.
For maintaining that balance.
In the central nervous system.
Absolutely.
But what happens if things get out of whack?
Well, imagine a car with only a gas pedal.
Okay.
And no brakes.
You'd be in for a wild ride.
A wild, uncontrolled ride.
Yeah.
The same is true for our brains.
Right.
Too much excitation
can lead to
problems like seizures or anxiety.
Too much inhibition
might cause lethargy or even coma.
So these GABA and glycine,
they're essential.
They keep things running smoothly.
They keep the activity in check.
So they're like the unsung heroes of our brain.
Absolutely.
Maintaining order amidst all the chaos.
That's a great way to put it.
But how do they relate to things like sedation?
Oh, that's a good question.
And anesthesia.
Yeah.
Where we want to dampen brain activity.
Right. So that's where the dose-dependent effects of GABAergic drugs come into play.
Okay.
These are medications that enhance the effects of GABA at its receptor. At low doses, they induce this sense of calm and relaxation, which is why your doctor might prescribe a benzodiazepine for anxiety.
But at higher doses?
At higher doses, they can lead to that profound inhibition we see in general anesthesia.
So it's like turning down the volume
of
brain activity.
It's not about shutting down the brain entirely, but more about enhancing that braking power of GABA.
Okay, making it harder for neurons to get overexcited.
Exactly. And this principle applies not just to anesthesia, but to a range of other conditions.
Like how?
Well, remember how we talked about the balance?
Between excitation and inhibition being so crucial for a healthy brain.
When that balance is disrupted,
it
can contribute to a variety of
neurological and psychiatric disorders.
For instance, too little inhibition
could play a role in conditions like
epilepsy, where you have abnormal bursts of electrical activity.
Leading to seizures. So are you saying that targeting these inhibitory pathways with drugs could help manage those conditions?
That's precisely what researchers are exploring.
That's amazing.
By understanding how GABA and glycine receptors work. And how different drugs can modulate their activity. We can develop more targeted therapies for a range of conditions.
Okay, for things like anxiety and insomnia.
Yes.
Epilepsy and chronic pain.
It's a truly exciting field of research.
Wow, it's amazing how these tiny receptors,
they are tiny,
these gatekeepers of chloride ions,
They are.
hold so much power
It's fascinating.
over our brain function and well-being.
What's even more fascinating is that GABA A receptors, they come in different subtypes.
So subtypes.
Each with a unique structure and function.
So they're like specialized versions
You could say that.
of those doorways, each tailored.
Tailored for a specific purpose.
Okay.
Some are particularly sensitive to certain drugs,
Okay.
while others play a role in specific brain circuits.
That makes sense. Because if all GABA receptors were the same,
Right.
wouldn't a drug like a benzodiazepine affect the entire brain equally?
Yes, but we know that's not the case.
Yeah.
That's why understanding these receptor subtypes
It's so important.
is crucial for developing drugs
With fewer side effects.
that are more targeted.
Imagine a drug
Imagine.
that could enhance GABAergic activity.
Specifically in the amygdala.
In the amygdala. Okay. A brain region involved in fear and anxiety
Exactly.
without affecting other areas of the brain.
Right.
That would be...
A game changer
Yeah.
for treating anxiety disorders.
Wow. You've officially blown my mind.
It's incredible, isn't it?
These GABA receptors are way more complex
They are.
and nuanced
There's so much more to them.
than I ever imagined.
And we've only just scratched the surface.
Wow, so much to explore about these fascinating receptors
There is
and their role in shaping everything from our mood
everything,
and sleep
our ability to learn.
to our ability to learn and remember.
It really is amazing.
But for now,
Yes.
let's take a step back and consider this.
Okay.
If GABA is primarily responsible for inhibition,
Yeah.
what about its counterpart,
Excitation.
excitation?
Yeah.
How does that fit into this intricate dance of brain activity?
That is a great question. You know, glutamate is the main excitatory neurotransmitter
Right.
in the brain.
So how do these two systems
The gas pedal and the brakes.
work together to create the symphony
The symphony.
of our thought,
Yeah.
feelings, and actions?
We'll delve into that in just a bit.
Awesome. Can't wait.
It's going to be good. It's like this perfectly choreographed dance. You have GABA and glycine are inhibitory stars putting on the brakes while glutamate, the excitatory leader, keeps the rhythm going.
So it's not a battle between good and evil,
No.
excitation versus inhibition. It's more about finding that sweet spot, that perfect balance.
Exactly. Think of it like a seesaw.
Yeah.
You need both sides to keep it balanced and functional.
Right.
Too much excitation, and the seesaw tips wildly in one direction, leading to chaos and potentially even damage. Too much inhibition, and the seesaw slams down on the other side, causing everything to grind to a halt.
That makes sense. So how does this delicate balance actually play out in the brain? We've talked about GABA and glycine dampening neuronal activity.
Yeah.
What about glutamate? How does it rev things up?
Well, glutamate binds to its own set of receptors on neurons, and these receptors are like the opposite of GABA and glycine receptors.
That's the opposite.
Instead of opening channels for chloride ions, they open channels for sodium ions.
And sodium ions are positive.
You got it. So when glutamate binds to its receptor, it triggers an influx of positive ions into the neuron.
Yeah.
This makes the inside of the neuron more positive, moving it closer to that firing threshold.
So it's giving the neuron a boost of energy,
Like stepping on the gas pedal.
while GABA and glycine are pumping the brakes.
Exactly.
And the interplay between these two systems determines the overall level of brain activity.
Precisely. It's a constant conversation, a back and forth between excitation and inhibition. And this conversation is happening all over the brain in different regions and at different times.
That's amazing.
It's what allows us to process information, form memories, make decisions, and experience the world around us.
It's mind-boggling to think about all of this happening at a microscopic level within billions of tiny neurons constantly firing and communicating with each other.
It truly is a marvel of nature. And speaking of marvels, let's talk about something that can directly impact this delicate balance: alcohol.
Okay, here we go. Everyone knows that alcohol has this like relaxing effect. Is that because it's messing with this glutamate GABA seesaw?
You bet. Alcohol actually enhances the effects of GABA at its receptor,
Oh.
like giving those brakes an extra push.
Okay.
At the same time, it can also interfere with glutamate's ability to bind to its receptors, essentially weakening the gas pedal.
So it's a double whammy. It's boosting inhibition and dampening excitation at the same time.
Precisely. And
Right.
that's why even small amounts of alcohol can have noticeable effects on our mood, behavior, and coordination. We feel more relaxed, less inhibited, and maybe a bit clumsier as our motor control gets a bit loose.
But there's a catch, right? We can't just keep hitting those brakes without consequences.
You're absolutely right. The brain is incredibly adaptable, and it doesn't like being pushed around if we keep flooding it with alcohol. It starts to compensate.
Compensate how?
It might start producing less GABA or become less sensitive to its effects.
Okay.
And on the glutamate side, it might increase the number of glutamate receptors or make them more sensitive.
So the brain is essentially trying to restore that balance, even if it means becoming dependent on alcohol to do so.
That's one of the reasons why alcohol can be so addictive over time. Those initial pleasurable effects, that sense of relaxation and lowered inhibitions, become harder to achieve.
Right, you need more.
The brain needs more and more alcohol to maintain that altered state.
It's a reminder that even though these inhibitory neurotransmitters are essential for a healthy brain, they can also be hijacked, leading to problems like addiction and dependence.
And that underscores the importance of moderation and responsible alcohol consumption.
Right.
We need to respect the delicate balance of our brains and avoid overwhelming those finely tuned systems.
So we've talked about alcohol, but what about other substances? Are there other drugs that target these GABA receptors?
Oh, definitely. There's a whole class of medications called GABAergic drugs, and they work in various ways to enhance the effects of GABA.
Okay.
We've already touched on benzodiazepines,
Right.
which are commonly prescribed for anxiety and insomnia.
Right.
They bind to a specific site on the GABA receptor, making it more receptive to GABA's calming effects.
So they're giving those breaks an extra boost.
Exactly. And because different benzodiazepines bind to slightly different sites on the GABA receptor, they have slightly different effects and uses.
Okay.
Some are better for anxiety, while others are more effective for sleep disorders.
That's fascinating. It's like we're fine-tuning those inhibitory signals, targeting specific areas or symptoms.
And the research continues. Scientists are constantly exploring new ways to target GABA receptors, searching for treatments for conditions like epilepsy, chronic pain, even neurodegenerative diseases like Alzheimer's.
It's incredible to think that these tiny receptors, these gatekeepers of chloride ions, hold so much potential for understanding and treating a wide range of conditions.
But what about the long-term effects of these drugs?
Yeah. Are there any downsides to chronically enhancing GABA activity?
That's a great question, and it's something that researchers are actively investigating. While GABAergic drugs can be incredibly helpful in the short-term, long-term use, can lead to tolerance, where the brain becomes less responsive to the drug's effects.
Okay.
And abruptly stopping these medications can sometimes lead to withdrawal symptoms, highlighting the importance of careful management and tapering under medical supervision.
It sounds like we're still learning a lot about the long-term implications of tinkering with these delicate systems.
Absolutely, but that's the beauty of science. We're constantly pushing the boundaries of knowledge, uncovering new insights, and refining our understanding of the brain's incredible complexity.
So we've covered a lot of ground today, from the intricate dance between excitation and inhibition to the potential and pitfalls of GABAergic drugs. But before we wrap things up, I have one more question. We've talked about how these receptors are like gateways controlling the flow of ions, but what about the structure of these receptors themselves? What makes them so unique and specialized?
That's a great segue into the next part of our deep dive. These receptors are not just simple doorways.
Okay.
They're complex protein structures with multiple subunits, each playing a specific role in their function. And understanding those subunits is key to unlocking the secrets of how these receptors work and how we can target them with drugs.
Okay, now you've really piqued my curiosity. I can't wait to delve into the structural details of these fascinating receptors. Let's take another deep breath and dive right in. Okay, so we're back.
Back again.
And you mentioned that these receptors are not just simple doorways.
Right.
But complex protein structures.
Precisely, think of them.
Like?
Elaborate locks, each with
Multiple keyholes.
multiple keyholes and levers.
Okay, so the keys are the neurotransmitters.
Exactly, but it's not just one key.
Right. Yeah.
These receptors are made up of 5 protein subunits.
Okay, 5 subunits.
And each subunit has a specific role to play.
So it's like a 5-piece puzzle.
Yeah.
That has to come together.
Perfectly.
For the receptor to work.
That's a great way to put it.
So these subunits,
All the same.
they're not all the same.
There are actually multiple types of subunits.
Okay.
Like alpha, beta, gamma, delta, and even rho.
Whoa, that's a lot of Greek letters.
It is.
Does each subunit have a specific job?
You bet they each have a slightly different structure and function.
Okay.
And the combination of subunits determines the receptor's overall properties.
Okay.
For example, some subunits are responsible for binding GABA,
Okay.
while others influence the sensitivity of the receptor to other molecules like benzodiazepines.
Feels like having
Different
different versions of those keyholes, each designed for a specific type of key.
Precisely. And this variability in subunit composition.
Is one of the reasons.
Is one of the
Why there's so many different subtypes.
of GABA receptors.
Okay.
Each with its own unique set of properties and roles in the brain.
But why is this important? Why should we care about these tiny subunits?
Well, understanding these subunits is crucial for developing drugs
Okay.
that are more targeted and effective.
Yeah.
Imagine if we could design a drug
That's specifically.
that specifically binds to a certain subunit combination,
Only in certain brain regions.
found only in the brain regions involved in anxiety.
Okay, yeah.
For example, that drug could potentially alleviate anxiety
Right.
without causing unwanted side effects.
In other parts of the brain.
In other parts of the brain.
That would be amazing.
It would be a game changer.
Like having a master key
A master key.
that only unlocks specific doors.
Exactly. And that's why researchers are so focused on mapping out these subunit combinations. Understanding their roles in different brain circuits.
Yeah.
And developing drugs that can selectively target them.
So we've gone from the big picture
From the big picture
of excitation and inhibition
down to the molecular level.
down to the molecular level of these intricate receptor subunits. It's incredible how much complexity is packed into these tiny structures.
It really is, and that's just the tip of the iceberg.
Oh, there's more.
We haven't even touched on the fascinating world of glycine receptors, which have their own set of subunits and complexity.
Oh, wow. So much to explore.
But for now,
Okay.
let's take a moment to appreciate the incredible journey we've taken today.
It's been amazing.
We've explored the crucial role of inhibitory neurotransmitters
Yes.
in maintaining a healthy brain,
Yes,
delved into the intricate mechanisms of GABA-A and glycine receptors,
we sure did.
and even glimpsed the future of drug development targeting these fascinating structures.
It's been an eye-opening deep dive.
It adds
We've seen how these tiny molecules
these gatekeepers of chloride ions
orchestrate this delicate balance
that shapes our thoughts.
that shapes our thoughts, feelings, and actions.
And we've learned that understanding these intricate systems is key to developing new and better treatments
For a wide range.
for a wide range of neurological and psychiatric conditions.
And while there's still so much more to uncover,
There is.
we've laid a solid foundation for understanding
We have.
this essential aspect of brain function.
As we continue to explore, we're bound to discover even more fascinating insights into the incredible complexity and adaptability of the human brain.
So keep those minds curious.
And keep exploring
There's a whole universe waiting to be discovered.
within each of us
Thanks for joining us on this incredible journey
into the world.
into the world of inhibitory neurotransmitters.
Until next time.
See, Shay,